Well, I prepared 2 lectures today. Seems that after chapter 3 and 4, you should automatically go to chapter 5. So you'll see, and then I checked the syllabus and remembered that I like to do it out of order to continue the discussion on neuropeptides before going to neurotransmitters. So you'll see that chapter 11 is posted, and that is the lecture covered in this recording. So I'll switch over to the slides. What I'm trying is a blue background with yellow letters. Wow. And we'll see how that projects. It comes in the small window anyway, so I don't know if it'll make a tremendous difference. But the copies posted on WebCT are black and white. I changed the format. So for your viewing and or printing, you should find that that's much more comfortable. But I thought I'd try a little bit of color just to test our system here. So in chapter 11, we begin the discussion of neuropeptides 1, classification, synthesis, and co-localization with classical neurotransmitters. And then chapter 12 will be the neuropeptides 2 in the next lecture. This slide contains table 5.1, which is covering neurotransmitters. And it lists the 8 criteria for determining whether or not a neuroregulatory chemical is a true neurotransmitter. And I'll read through those in just a moment because we're considering neuropeptides in the context of our discussion in this lecture more so as acting transsynaptically as neurocrine molecules throughout the nervous system. But also we are keeping in mind the neuropeptides from the hypothalamus that are part of the neuroendocrine function. So we kind of swap concepts across the board, neurotransmitters, neuromodulators as neuroregulators, and the neurohormones acting in a typical endocrine type of a function from the hypothalamus to the pituitary or posterior hypothalamus to the target organ in the body. So the neuropeptide is any hormonal or non-hormonal peptide that acts as a neuroregulator, either a neuromodulator or a neurotransmitter. And our challenge in this course, as we've always said, is to start from the textbook, then we see definitions and distinctions, comparisons and contrasts, and generalizations. Then we get to the literature and we look at the nature of how a peptide will act on a neuron, and it'll behave very much like a neurotransmitter. So let me just read through this list of 8 things. The substance must be present in presynaptic neurons, usually in an uneven distribution throughout the brain. That is, certain brain areas will make one neurotransmitter, whereas others will make an alternate one. This is indeed true for neuropeptides. Neurotransmitter precursors and synthetic enzymes must be present in the neuron, usually in close proximity to the site of action. Altering this to consider the synthetic mechanisms, biosynthetic mechanisms at the cellular level for neuropeptides. This is also satisfied, and the synthesis is one of the things, synthesis of peptides, neuropeptides, is one of the concepts we'll consider in this lecture. Stimulation of nerve afferents, the dendrites, should cause release of the substance in physiologically significant amounts. This indeed has been demonstrated for neuropeptides as for classical transmitters, neurotransmitters. The effects of direct application of the substance to the synapse should be identical to those produced by stimulating afferent nerves. There's a portion in this chapter on neuropeptide agonists and antagonists, and indeed we'll look at electrophysiology throughout the middle part of our lecture series this semester, demonstrating the similar type of thing, either of exogenous peptide that's identical to the endogenous ligand or exogenous analogs that might be modified to delay their degradation or indeed be non-peptide molecules that act at the receptors. So this is satisfied, can be satisfied. Interaction of the substance with its receptors should induce changes in postsynaptic membrane permeability, leading to excitatory or inhibitory postsynaptic potentials in the postsynaptic cell. And this indeed can be satisfied with peptides, as you will see. There are some deviations from this statement that demonstrate, in my mind, demonstrate the definition of a neuromodulator very nicely, and we'll see that in the next lecture, and then one of the subsequent lectures focusing on the neurophysiology as well. Specific inactivating mechanisms should exist which stop interactions of the substance with its receptor in a physiologically reasonable time frame. We'll talk about neuropeptide degradation today and see that the appropriate degradative enzymes, peptidases, exist within the extracellular interstitial fluid of the nervous system, basically everywhere. I don't know if they're concentrated at synapses, but available near synapses equally as anywhere else to degrade the neuropeptides. Interventions at postsynaptic sites using agonist drugs should mimic the action of the transmitter, and antagonists should block its effects. So it's essential to demonstrate a receptor-dependent mechanism for the postsynaptic or target cell effect. So again, we have to keep the idea of a textbook in mind, and then remind ourselves that it's the specifics that matter. So whether any of these classical transmitters satisfy all 8 of those or not, we consider them as neurotransmitters, and indeed all of them do satisfy that. Less is known about histamine than these others, the serotonin, glutamate, GABA or gamma aminobutyric acid, noradrenaline and adrenaline, dopamine and acetylcholine. We know that some of these also can act as neuromodulators, modulating the response of target neurons or target cells in their response to some other chemical stimulus. Now, so it's kind of a challenging thing to create these strict definitions in biology, especially cellular communication mechanisms, and try to live by them. So I pulled another table from another source, the hypothalamic neurohormones, rather than just having the one from our textbook, to show some of the complex interactions of neurohormone or neuropeptides from the hypothalamus targeting the anterior pituitary, the hypophysiotrophic hypothalamic neurohormones. So there's thyrotropin-releasing hormone, or TRH. It's a peptide with 3 amino acids, so it's really a small one. Still, I think it holds that position as the smallest. Gonadotropin-releasing hormone, or G lowercase nRH, has 10 amino acid residues as a peptide. Dopamine, we know, is about genic-amine, as we'll talk about. The corticotropin-releasing hormone, or CRH, is 41 amino acid residues. Growth hormone-releasing hormone, GHRH, or you may even see on an exam, a test, GRH. If it's not G lowercase nRH, and it's just GRH, you assume, therefore, that it is growth hormone-releasing hormone, not the gonadotropin-releasing hormone. Another name for GnRH is LHRH that you might see in the literature or textbooks. That's luteinizing hormone, releasing hormone. For some reason early on, that hypothalamic releasing hormone got that name tagged onto it, and then they made it more generic because it was thought that there's a follicle, an FSHRH as well as an LHRH, and turns out there's a single GnRH that is the prime secretagogue for the gonadotropins. And then somatostatin. So growth hormone releasing hormone is also called somatoliberin, Liberin, to liberate, right, in the nomenclature of somatostatin. So technically then it would be growth hormone releasing hormone, growth hormone inhibiting hormone, and somatolibrin with somatostatin, to be very picky about it, right? So we see that the number of amino acid residues is increasing up to the GHRH 40 to 44 and back down to 14 for the somatostatin. So up to 10 amino acids, and I know the nomenclature for 12, the dodeca, but this is a decapeptide and tripeptide. Once you get past the decapeptides, basically you refer to the number of amino acid residues for the molecule. So the TRH obviously is going to cause the release of TSH, but also stimulate, and again this is synthesis and secretion. Whenever we think about stimulating release, we automatically extend that in our minds to synthesis and secretion. You secrete it, you have to synthesize to replace what you released. So it also affects prolactin synthesis and secretion. GnRH, obviously LH and FSH, but some question as to whether it affects the prolactin as well. The asterisk here is that under physiological conditions and when administered exogenously in intermittent pulses. So the GnRH is a fun molecule to work with in the classroom context, especially getting to an exam, because if it's administered exogenously at constant levels, it will inhibit the anterior pituitary, and we'll talk about the molecular mechanisms of that at the end of the semester. If it's given in a pulsatile fashion, then it sustains the anterior pituitary function for the gonadotropins. Turned out to be very useful discovery, and the scientists that discovered GnRH, thought if they injected themselves with it, they'd become some reproductive physiological god, to put it in polite terms or something. So they were injecting themselves with it, and they found that they had an inhibition of LH and FSH, and actually the opposite effect of what they fantasized or hypothesized, whichever it was. And so that discovery, serendipitous discovery, actually turned out to be very clinically relevant, especially for suppressing the pituitary in patients that suffer from endometriosis. And we'll bring that up again when we talk about the HPG axis. But just to follow through with that asterisk footnote there, I did want to bring it to your attention. So if you see a graphical display of GnRH, it should be pulsatile. If it isn't, then something is wrong. The dopamine is inhibiting prolactin. It is the only known, fully identified and characterized prolactin inhibiting hormone, although they're suspecting a peptide one. It still is not finalized yet. But it also can have effects on LH and FSH, the gonadotropins, their release, and thyroid stimulating hormone. Corticotropin releasing hormone, the CRH, obviously targets ACTH. We'll talk about the gene for ACTH and the synthesis and processing, and obviously it is the releasing hormone, so it's going to stimulate. GHRH, obviously going to stimulate synthesis and secretion of growth hormone, and somatostatin, targeting not only the somatotropes, but also the thyrotropes, and inhibiting the release of their cognate hormones, GH and TSH, respectively. Continuing on with the concept of peptides as chemical messengers, there's obviously many other families. I'm presenting this illustration in contrast to what's in the text, but either complements, supplements, or matches. It's not a great concern. Always presenting to you exactly what's in the textbook isn't really that fun. So the peptide families, there's gastrin and CCK families, including those two. Secretin, glucagon, VIP, with other molecules like PHI, GIP, PACAP, GLP-1, 7 through 36. So you can see that there's a large number of peptides, and some of them only contain certain amino acid sequences of the full peptide from which they were generated. So it's not so much that I want you to know all the names for these acronyms as it is to recognize the plethora of peptides that are acting in biological organisms as chemical messengers. Pancreatic polypeptides, there's the pancreatic polypeptide itself, a neuropeptide Y and a peptide YY. We'll hear a little bit about neuropeptide Y as we're going through our discussions. We've already heard about CCK. Pertinent to today's lecture, is presented as being co-localized with dopamine within the motor and reward and emotion circuits in the limbic system. Others are GRP, motilin, galanin, neurotensin, somatostatin, and we'll see just a couple of others today. These peptides can be categorized as endocrine, neurocrine, or paracrine substances. And so the endocrine ones are somatostatin, cholecystokinin or CCK, gastrin, secretin, insulin, glucagon, enteroglucagon, pancreatic polypeptide, the ones acting in a neurocrine fashion, relevant to our other consideration today, being able to act within the nervous system, synthesized and secreted within the nervous system, acting transsynaptically or extrasynaptically, to function in the neurocrine or even paracrine sense as far as that's concerned. So the somatostatin is in both lists, CCK is in both lists. There's a GRP, we're not going to worry about acronyms we don't recognize. The opioids, substance P, VIP. This is vasoactive intestinal peptide, so obviously it was discovered first somewhere else in the body, in the intestine, within neurons there, and having effects on the local vasculature, and then discovered as to being expressed in neurons within the central nervous system as well, and having significant effects. Neuropeptide Y we saw under the pancreatic list, also within neurons, and neurotensin. And then those acting in a paracrine fashion, where it's established, the somatostatin and the peptide YY. So we had talked about these definitions in general, and we just present some examples in this case. You know, still just taking this general approach of getting an overview and appreciation of neuroendocrinology and endocrinology in general. So continuing, neurotensin, also an endocrine peptide, motilin, glucose-dependent insulinotropic peptide, GIP, peptide YY, urogastrone, epidermal growth factor. So just tons of names, pancreastatin, galanin, motilin, and peptide YY, also acting within neurons. So that's our rather mundane but also required reading of the list of molecules, presenting terms so we hear them and see them and begin to build some familiarity and ability to recognize them and place them into context. Now we can move on to something that's a little more engaging of our prior knowledge and prior lecture with this very nice illustration. So we want to think now about the synthesis of peptides, or the smaller protein types of hormones, the polypeptides it would be. And we know it comes back to the genes. So we have in the upper panel an illustration of the setup of a gene on a strand of DNA, double helix DNA, with RNA polymerase II right nearby, with a promoter region that we've illustrated and talked about. There's also a terminator region. You've got to stop at the end of the gene, right? And then we can talk about initiation, elongation, and termination just briefly. So the RNA polymerase somehow has to bind to the start site. Tatabox is involved in that on the promoter region, as we know. Starts with the first nucleic acid and nucleotide, to the right, we want to think of it that way, in the positive or downstream to the start site, and begins to assemble the RNA such that the starts reading DNA on the 3' end and produces the 5' end of the RNA strand. So that is the initiation. When we start talking in more detail about regulation of gene expression and the factors that are involved, we consider the promoter region in greater detail. What I like about this illustration is it shows the uncoiling of the DNA beyond what the histones may have done. Once you get the histones to free it up, so you've got euchromatin, it's still the double helix DNA, so something has to come in to relax that helix and allow all these molecules to get in there, especially at the start site where the RNA polymerase II has to bind. So transcription factors that can relax this helix more or tighten it up either increase or decrease the probability that RNA polymerase II will be able to bind to the start site on that gene. So it helps us to visualize that. And then it moves along, and as it's reading the genetic code, as we know, introns and exons, it produces the full length or the nascent strand of RNA, in this case the growing chain. 5' cap is added very quickly and persists, and when it is reaches the termination site, of course the RNA polymerase II falls off, as well as the primary transcript or the nascent strand of RNA is then available for additional RNA processing. The splicing is not shown here, of course, but the poly A tail addition is, and then the nuclear pores, as we know, are available once the poly A tail and the 5' cap are on, are available to provide permission for this mature messenger RNA, as we're considering, to exit. So that's just that quick reminder of that information for peptides is extremely important. So when we talk about neurotransmitters that increase the release of a hypothalamic neurohormone, we link it to synthesis. Somehow the transmitter signal with its transmembrane signal transduction that's dependent upon its receptor, we're thinking of water-soluble chemical messengers now, must diverge in one way, changing membrane properties, affecting firing rate, therefore release, and in the other pathway, diverge off and get a signal to the nucleus that can target the gene for the biosynthesis of that peptide and begin the process of replacing what was lost, or well, what was secreted, not really lost, right? So this is figure 11.1 from our text, compares the classical transmitter synthesis, which we will talk about when we get to chapter 5 after doing neuropeptides 2, but there's no harm in seeing this. We've all thought about this a bit before, and comparing that to the neuropeptides. So I'll start with the neuropeptides and then talk about the neurotransmitters. So this is considering the neuronal synthesis and secretion of CCK. So we just talked about what happens in the nucleus to get the message out to the rough ER, and then something happens there and along the way in the transport vesicles to the Golgi, which finally packages it, of course, into the secretory vesicles or granules in the neuron cell body or soma, that's transported anterogradely to the terminal, the axon terminal, where release will occur, and we'll discuss this in more and more detail today than before, and then more detail as we proceed through the semester. So the production is first of a polypeptide precursor. So we talked about translation and post-translational processing, so we'll look at some examples of that today. There might be conversion enzymes that are involved, like cleavage enzymes. We'll give a couple examples today. And then, of course, on to the secretory vesicles, the axonal transport, and then the supply is regulated by the axonal anterograde transport and storage. So in other words, there's no local synthesis or recycling. It has not been established that there is recycling yet of neuropeptides in the terminal. The gene transcription is the sole source of generating these neuropeptides directly. So the messenger RNA produces the chemical messenger, and then that has to get to the site of release before it can have a biological effect. Neurotransmitters, on the other hand, are synthesized from precursors and accomplished, and that is accomplished by biosynthetic enzymes in a biochemical or biosynthetic cascade or pathway. And we'll talk about some of those in the lectures on neurotransmitters. So in this case, for a transmitter, we're thinking about genes that are expressed within a neuron that have as a gene product the synthetic enzymes that are required for this series of biochemical steps or changes in the biosynthetic pathway. So we have to start getting this in our mind, because when we talk about the steroidal synthesis, it's the same thing. Cholesterol is converted from this to that, and then the intermediate is converted from this to that. So we all remember some of that from biochemistry or whatever course maybe we have taken where we've seen these. And when we talk about the classical neurotransmitters, the catecholamines, we'll look at the actual chemical structures and talk about the enzymes and their cofactors and get into the biochemistry a little bit. So it's the genes for those biosynthetic enzymes that are whose expression are being regulated when we talk about coupling release of a transmitter with its synthesis at the level of the gene. Because every enzyme has a life cycle too. It doesn't live forever, so it will disappear. So we have to think of the gene for the biosynthetic enzyme being expressed such that the gene product for that enzyme ends up in the vesicle as well. And typically in the cell body, the vesicles subsequent to the Golgi already have the biosynthetic capability. Now sometimes there's cytosolic biosynthetic enzymes that interplay with the intermediates that end up in the vesicle, and we'll see examples of that. So both would have to be there. Both the cytosolic and the intravesicular enzyme systems would have to be there, and appropriate mechanisms to shuttle the intermediate or final transmitters in one direction or another in and out of the vesicle to accomplish that biosynthesis. But biosynthesis of the biologically active neurotransmitter is possible at the level of the vesicle upon leaving the Golgi. So it is possible, and indeed it's even possible for the neurotransmitter to be released at the cell body and the dendrites. Serotonin is a prime example of that, as well as dopamine. So anyway, it's a packet of biosynthetic machinery that is sent down the axon terminal, down to the axon terminal with anterograde axonal transport for the neurotransmitters. And the neurotransmitter can be synthesized along the way as that vesicle is recycled with release and uptake, so exocytotic and endocytotic types of events, considering the membrane itself. Recycle the vesicles, and the biosynthetic machinery persists, so you can have synthesis of neurotransmitters at the terminal, but you do not have that occurring for neuropeptides. So the supply for the neurotransmitter at the terminal is regulated by the axonal transport, as well as vesicular storage, new synthesis, and reuptake, as we'll see repeatedly. So this is a primary distinction. The mechanism, the types of genes and gene products that are present, and the mechanism for synthesis, and then the distinctions at the axon terminal, the site of release. How is the pool, the releasable pool, sustained? I found another site that this is from Endotext, but there's another site from the ACNP, the American College of Psychopharmacology, ACNP, American College of Neuropsychopharmacology, and that site is listed. I didn't put it on the home page. I put it under the course content. Is that the folder on the home page? You'll see the link there. It's a fantastic online resource, and we'll be referring to it throughout our series of lectures, especially on the methods. But I have some information from that as well that definitely expands upon information within the textbook. So now we're thinking about transcription, translation, and post-translational processing, and we can use antidiuretic hormone or vasopressin as the example. I like this illustration because it reminds us of the gene having exons and introns. So the vasopressin gene, it has 3 exons, and we see those exons represented in the pre-pro hormone, the vasopressin neurophysin 2 precursor, the amino terminal on the left, carboxy terminal on the left, on the right, and we see all these different parts along the way. Represented by the N-terminal end representing exon 1, exon 2 in the center for the neurophysin 2, and exon 3 represented there. These little arrowheads obviously are showing potential cleavage sites, and typically those are lysine-lysine or lysine-arginine residues, as we'll talk about as we go along. So particular cleavage enzymes have to target those. These would be endopeptidases, right? If they act anywhere within the peptide, they are endopeptidases. If they started at the end and worked their way in, they would be exopeptidases. So do we remember all of our basic biochemistry? So the structural organization of the vasopressin neurophysin 2 gene and processing of its product is shown here. It's got 3 exons. Translation of the messenger RNA yields a large pre-pro hormone precursor, subsequently modified through substantial post-translational modifications, and we'll see more of those as we proceed. The oxytocin gene, remember it differs by only a couple of amino acid residues, has a similar structure, and its product undergoes similar processing and post-translational modification. So this is from our Endotext source. So we see that the one gene for vasopressin contains the sequences within exon 1 for 1 copy of the vasopressin hormone. We'll see in other examples where in the one gene distributed throughout the exons, there will be multiple copies of the hormones, and that's for thyrotropin releasing hormone, which is only the tripeptide, so that's very possible. So the synthesis of the vasopressin oxytocin precursors occurs in the cell bodies, again, in the soma, as we talked about, of the specific magnocellular neurosecretory neurons, but there are other neurons distributed throughout the brain that aren't big ones that express this gene and release it at synapses to have effects. So it's the supraoptic nucleus as well as the paraventricular nucleus. And the generation of the mature hormone entails the post-translational modifications of the large primary precursor, as we had seen, and we'll look at in more detail in the next figure. And then after the translation, the carboxy terminal domain of the precursor is glycosylated, so there's glycosylation, and the product is packaged in the vesicles of the regulated secretory pathway. So these vesicles will migrate along the axons, as we said, down to the posterior pituitary. And during that migration, the precursor is cleaved by basic endopeptidases. So basic referring to the basic amino acid residues, lysine, 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 arginine, the endopeptidase as described, into the mature hormone and the associated neurophysin. And they're stored in the secretory granules in the posterior pituitary. And when there's increased firing rate, there's voltage-gated calcium influx, calcium channels that allow the influx with the classic type of exocytotic release. So that leads to that transient calcium influx, and the granules move to the presynaptic membrane, as we know, and we get the exocytotic release of the mature biologically active vasopressin or oxytocin and its corresponding neurophysin into the systemic circulation in equimolar quantities. So in other words, for every vasopressin or oxytocin molecule that's released, there's one neurophysin molecule that is released. So there's a 1-to-1 ratio or equimolar quantities of these molecules in the blood. So the neurophycins will act as the carriers for their cognate hormones, neurophysin 1 for oxytocin, neurophysin 2 for vasopressin, during the axonal migration, and there still is no other function that's been discovered for them. So they're not functioning as carriers for these hormones in the plasma. No one knows exactly what they are doing. And it is a bit bizarre because we don't think, when we think of conservation of energy, we don't think that any living cells waste energy on producing proteins that don't have a function. And the meager function of just being there in the vesicle to get the molecule to the terminal for release just doesn't seem to be significant enough for this large piece of protein. But biology fools us. So this is figure 4. I'll read the legend here real quick. It's a schematic overview of the post-translational processing of the vasopressin neurophysin gene product, again with more detail. There's a sequential modification of the NP2 pre-prohormone in the endoplasmic reticulum and the Golgi. So the post-translational processing is initiated in the ER and continues in the Golgi, ultimately trafficking through the regulated secretory pathway and then ultimately released from the neurosecretory vesicles in the posterior pituitary, as we said. Small amount of partially processed precursor is released through the constitutive secretory pathway, and oxytocin is processed in a similar manner. So again, here we see the messenger RNA for the vasopressin neurophysin 2. Here's the vasopressin from exon 1 and the neurophysin 2. And then this CP, the C-terminal peptide, and the signal peptide. So the messenger RNA meets up with the ribosomes, as we know. Translation, targeting, and co-translocation occurs within the ER, as we know. So within the ER, there's clipping of the signal peptide. Signal peptide is essential for informing the synthetic conglomerate, if you will, the ribosomes basically, that this is a secretory hormone that must be synthesized and put into the ER simultaneously. So the first event is clipping or cleavage of the signal peptide, and then we see the prohormone here, and there's glycosylation of the C-terminal peptide or portion of it, the CP, and single glycosylation event in the endoplasmic reticulum. So after the signal peptide is released, it's 18 kilodaltons. Add the sugar side chain, and it goes up to 21 kilodaltons. Now, there's also a disulfide bridging and folding. We saw that with the illustration in the previous lecture for the oxytocin and vasopressin. And then when it reaches the cis-Golgi pathway, there's a second glycosylation event on the C-peptide, C-terminal peptide, bringing it up to about 24 kilodaltons. And that is the form that is secreted in a constitutive pathway, just constantly being sent to the terminal to sustain a releasable pool. And then the regulated pathway that is dependent upon the signals to the neuron and the changing, or the alteration of the firing rate of the neuron. So this prohormone is then cleaved to produce the neurophysin carrying the vasopressin. It dimerizes and is packaged as it proceeds to the trans-Golgi portion, and the glycosylated C-terminal peptide goes along with it. It all gets packaged into vesicles. And so here we see the neurophysin 2 acting actually as a transport molecule for the vasopressin. And then with the axonal transport and with exocytosis, it just progresses as it approaches the axon terminal and can be occurring at exocytosis as well. You get the final cleavage so that you have the vasopressin and the neurophysin 2. The neurophysin 2 is 10 kilodaltons. Well, there's another gene that we can talk about in gene product, and that's discussed in the text as well. And this is in our text as well as endotext, and I've taken information from there. This is the pro-opiomelanocortin gene and gene product that undergoes post-translational processing, but produces multiple biologically active hormones. So we used one example for the posterior pituitary, oxytocin and vasopressin with their neurophysin and neurophysin 1 and 2 respectively, where the neurophisins were a cleavage byproduct but really had no biological activity, maybe some type of physiological activity as far as being a carrier, but that's not what we think of when we think of a biological effect acting at receptors and producing cellular changes. With the POMC as an example, the possibility of producing multiple different kinds of hormones that have cognate receptors that are unique and unique target cells and biological effects and activities. So it's an extension of that, but still the post-translational processing is, in essence, very much the same. So the ACTH is derived from a 266 amino acid precursor, and you begin to ask yourself, am I going to have to remember these numbers? Well, more and more they'll be popping up as a graduate level course. There are certain things that you'll be asked to remember, as far as molecular weights and the lengths. It won't be so much the 266 amino acid precursor, maybe in a multiple choice type thing, but whether one molecule is an octopeptide or a dectapeptide and have to spell that right and type it in yourself for the exam for the test is an example. So we'll build on that type of a thing of memorizing these details that's so typical of graduate courses as we approach exam 3, 4, 5, and so on. So the pro-opiomelanocortin is encoded by a single copy gene on chromosome 2p23.3. I'll never ask you to remember this, but if the chromosome number is included, sometimes I'll ask you to remember that information. Over about 8 kilobases. So it's got a 5' promoter, 3 exons, and apart from the hydrophobic signal peptide, we just talked about that in its cleavage, targets the ER for release. 18 amino acids of the N-terminal glycopeptide. All the rest of POMC is encoded by the 833 base pair exon 3. So the majority of POMC is encoded in its exon 3. So here we see a rather simplified view of the POMC gene product and its processing. Again, we see the potential cleavage sites as these dark vertical lines. Oh, just at the end of the time here. And the various fragments that can be produced from them. So we're at the end of our 50 minutes. I will start with this slide and continue on to the more detailed discussion in the next lecture. I appreciate your time and attention, and hope you have a nice day. Bye-bye.